This morning to introduce a very good friend, Professor David Mooney from Harvard University. Most of you probably know David. He's one of the most recognized and well-respected thought leaders in biomaterials and tissue engineering worldwide. Talking this morning, he said he doesn't like to be called distinguished because that makes him feel very prestigious. Although he is older than me. But the fact is that he has already had a very distinguished career, starting at the University of Michigan, getting his PhD in chemical engineering from MIT. And then he moved to Harvard, where he's the Robert P. Pincus Family Professor of Bioengineering in the Harvard School of Engineering and Applied Sciences. He's also a board faculty member of the Weiss Institute for Biologically Inspired Engineering. David is a member of the National Academy of Engineering. He's also chair-elect of the American Institute for Medical and Biological Engineering, or AMBE. He's an NIH Merit Awardee, and additionally he received an NSF Career Award. He's had several awards from different societies, including the Clemson Award from the Society for Biomaterials, an Einstein Visiting Fellowship, and several distinguished, again that word again, distinguished scientist awards from various societies, including the Tissue Engineering and Regenerative Medicine International Society of Troops. Now, David's research that you'll hear about today is focused on the design and synthesis of microenvironments, or niches, that regulate the function of either transplanted cells or cells that are already resident within the body. The biomaterial systems that he is developing mimic certain aspects of the native extracellular matrix and provide signals that may be useful for regulating the function of different stem cell populations. As you see, these systems can be applied to an extremely broad array of different applications and grand challenges related to regenerating tissues that have been damaged by disease or injury, treating autoimmune disorders, and targeting the destruction of undesirable tissue masses in cancer patients. As you'll hear, his work spans from fundamental discovery to technology development to translation to clinical applications. And his inventions have been licensed by numerous biotech companies, and he's involved in at least one that I know of, a very exciting clinical trial that I think you'll hear a little bit about today. So please join me this morning in welcoming Dr. David Mooney as a 2012 PhD Distinguished Lecturer.
Are you live? Yes, I should be live. Thank you. Thank you, Bob, for that wonderful introduction. I actually realized I should stop giving my own talks and let Bob give them, because he does it better than me. So it is really a pleasure to be here today, to be in Georgia Tech, which is the home for so much pioneering research in the tissue engineering and regenerative medicine area. I've really enjoyed my visit, learning more about all the different facets, well, I shouldn't say all, but many of the facets of the research that's underway here, and I look forward to meeting with some more of you this afternoon. So I'm going to talk about biomaterials, which is a focal point of the research in our laboratory. And before I get started, though, I want to just make some acknowledgments that the work I'm going to describe today would not be possible without some really wonderful collaborators, and we've had funding from a variety of different sources for the research that I'll describe today. Now, my starting point is going to be this very general picture. I think probably everybody in this room believes that we are now entering a new era in medicine, in which stem cells and cells more broadly will be therapeutic agents that are capable of treating a wide variety of different diseases that afflict millions of Americans. So I think, kind of the focal point of this institute is really based on this concept, and many of us obviously fervently believe this. However, if we think about down the road, when we've solved all the challenges that are inherent to using cells as therapeutic agents, where will that lead us? Where will we be at that point in time? Well, where will we be if we continue down our current path is with an industry that looks something like this, where we will have individuals in clean rooms wearing bunny suits culturing lots and lots of flasks of cells, very typically in a patient-specific manner. So this is what the industry that we would create might look like. And I think many of us can appreciate that this has some potentially really significant limitations. This would be an incredibly expensive way to make new therapies. The cell therapies that are out there today typically run, let's say, in the range of $100,000 per patient. It's very complex processes a tremendous amount of regulation, particularly when we're doing patient-by-patient manufacturing. And so the general idea of what I'm going to talk about today is a concept that we are pursuing to replace this vision of the future of cell therapies with this type of vision for the future of cell therapies, where instead of doing a lot of manipulation of cells in a laboratory, instead we design materials. In our hands, typically plastics, polymers, that do all the biology of interest in the body. If we have cells that are amenable to doing some of the things we're interested in that already exist in the body, why remove them? Why not instead try to manipulate them appropriately in situ? And so that's what I'm going to talk about today. And to give you a little bit more sense of this concept, this is just a schematic showing what we hope, in a very general sense, these material systems might do. So this blue region is supposed to be the biomaterial. And the concept here is we have some cell population that we think might be therapeutically useful. So we will release what I'm generically calling recruiting factors from the biomaterial. They will diffuse away from the material. They will bind and interact with that target cell population and induce those cells to move into the material. And the goal of having the cells live within the material is we now create a microenvironment in which we have tremendous control. So if we have, let's say, only a few cells that we can have available to us, we could give these cells appropriate signals to make them multiply within the device, to generate a large cell number. We may differentiate these cells or otherwise change them within the material, as indicated by this transition from this rounded cell to this kind of starfish-shaped cell. So generally, we're interested in programming the cells within this device. And then we may want those cells to act very locally, right around the material, or we may want those cells to travel to some distant location in the body. So we may need to target them to some site of action. And so the whole talk today is going to be based around this idea. So I'm just going to give you a few examples. And I'm going to start out with a couple of examples of how we're trying to recruit cells. And this goes back to work we've been doing for a number of years in the angiogenesis area. I'll talk a little bit about how we get cells to leave. So if we have cells in a material, how do we get them to leave? And can that be useful? And both those first two examples are going to be in the context of regenerative medicine types of applications. Now, what I'm going to skip today is I'm actually not going to talk a whole lot about programming the cells inside the material. I gave a seminar in the chemical engineering department yesterday where we really focused on that aspect, so I'm going to avoid being redundant and not talk about that today. But then, the last part of the talk, I'm actually going to try to bring this all together and talk about how we can, in the context of immunology, target a cell population, program a cell, send it someplace else, and get some biological function. So, pretty simple story today. I'll just kind of walk through this in stages. So the first step is recruitment. So we'd like to be able to, on demand, recruit cells from someplace else to a desired location in the body. And we've been exploring this in the context of vascular therapies, because as many of you are aware, cardiovascular disease, major cause of death in the Western world, and very frequently these diseases are caused by blockages of blood vessels that lead to downstream ischemia and death of cells and tissues. And just a few examples here of where this is prevalent. Now, one approach to try to deal with these vascular deficiencies is to try to build new networks of blood vessels by recruiting vascular cells into the tissue of interest to then return blood flow. And obviously the motivation here is very straightforward. If we don't have capillaries distributed throughout tissue, you rapidly get hypoxia as cells are removed from the capillaries and they will die. So what we'd like to do is recruit cells. And the process that we use as our inspiration is a normal process of blood vessel formation called angiogenesis that happens both during development and in the adult. And so the idea here is if you have a blood vessel, you have endothelial cells and then smooth muscle cells associated with those endothelial cells. If you need, if your body, for example, becomes hypoxic and cells over here don't have enough oxygen, they will secrete signals that will diffuse, bind to the endothelial cells, and activate those endothelial cells. So they will begin to migrate and proliferate. So these cells will be recruited. They'll ultimately form a new branch of a blood vessel that can carry blood and return perfusion to an ischemic region. Now this happens naturally to quite a limited extent, not to a sufficient extent to treat many of those diseases that I just talked a few minutes ago. So what we'd like to do is be able to direct this process and more or less have angiogenesis on demand, when and where we want it. And in particular, I'm going to use as an example the action of a particular growth factor called vascular endothelial growth factor, or VEGF, as our model system, at least in the first bit of the story, to talk about how we can use this molecule, which has been well identified as a potent activator and chemotactic agent for these endothelial cells to control migration. Now, some of you may be aware that actually this molecule has been widely explored in clinical trials in the past for therapeutic angiogenesis, and those trials failed, so they did not show the intended benefit. So, of course, the question then becomes, well, why did those therapies fail, and can we do better? Well, the premise of the work that I'm going to describe is that if we really want to get utility of a molecule like VEGF or a cell, a stem cell, or other agents, it's not just the act of injecting it into the body and then we're done, but it really is how we use it quantitatively that oftentimes is as important or perhaps more important. So in this context, when we started going down this path, we said, well, we would like to use this VEGF. We need to understand how to use it. So how should we use this to try to recruit cells when we want them to be recruited? Now, in essence, what we're trying to do is, from an engineering perspective, generate some design criteria for how we use this molecule. Now, we could do all this in vitro, or excuse me, we could do it all in vivo. That becomes relatively cumbersome and complex. So instead, what we typically do is utilize three-dimensional cell culture models that capture some key aspect of the biology, and then use that to understand the relation, for example, in this context, with some work done by Ruth Chen, a former PhD student, what is the relationship between endothelial cell sprouting in vitro, which is the model system we're using to, that we're using to mimic angiogenesis in vivo, and presentation of VEGF. And so this is endothelial cells that are moving off this bead in a process that's very similar to sprouting angiogenesis in vivo. And what Ruth showed was that VEGF is critically important to this process. If we present VEGF in different concentrations, we can indeed drive a lot of this sprouting behavior. However, there's an optimal concentration. Too little and we get no effect. Too much and we actually saturate the receptors and downregulate the response. Similarly, we typically want to recruit the cells to go in a specific direction. And to do that, we need to provide directional information. So we provide this molecule in a gradient, a spatial gradient. And what she's able to demonstrate is there's some minimal gradient that you need to present if you want to control what direction these cells go into. So you can start to develop general design criteria for recruiting cells with this general type of approach. And then, of course, we need to be able to actually achieve these concentrations and these gradients in vivo. And so the approach that we typically take is to put the appropriate biomolecule inside a polymer that will release it in a sustained and controllable manner at that site. So the next series of slides I'll show you involve use of injectable hydrogels fabricated from a polymer called alginate. It's a polysaccharide that we can gel by ionic cross-linking with divalent cations like calcium. We can inject this in a minimally invasive manner into tissues, as indicated here in this gel that contains a blue dye inside of tissue. And these are nanoporous materials, so we can encapsulate proteins and other biologic agents inside these pores, where we can control the time frame at which they get released. And we've documented all this in the past in a series of publications. I'll just show you one piece of data to demonstrate the idea here. So we can take one of these gels, we can load it with VEGF, we can inject it in a tissue, for example, right at this black dot, and this is a hind limb of a mouse. We can then sample the tissue in the surrounding regions, regions A, B, and C, and we can see how much VEGF we get in those tissues from the single injection. And what you can appreciate is we get about 2 weeks of exposure to this potent factor in the concentrations and gradients that our in vitro studies demonstrated were necessary to drive endothelial cell migration. And as a side note, we're able to accomplish this with having very little systemic exposure. So if we have an injection of a bolus amount of the protein in solution, it disappears quickly from the site. A lot of it makes it into the serum, so it's seen by the whole body. When we do the gel delivery, most of it stays very local. We get long-term presentation, and very little of it is seen by the rest of the body. So can this then help us to build networks of blood vessels? Well, if we take these gels and we inject them, this is actually just some marker gene. We're looking for expression of the receptor for VEGF, VEGFR2, and this just shows us that we have biologic activity for over 2 weeks when we inject these gels in the hind limbs of these mice. More importantly, if we actually look to see do we recruit endothelial cells and build capillaries, we can see that if we quantify the density of capillaries in the hind limbs of mice that were made ischemic, if we have all the controls, basically don't do anything, have VEGF in solution, so don't present it correctly, have the polymer by itself, we have no effect, But if we present the protein in the way that our in vitro studies describe to us or suggested to us that we should be able to recruit cells, we can now build networks of capillaries. And not only do we build networks of capillaries, these blood vessels, if we've built them correctly, have significant function. Here we're looking at the amount of blood flow, where we normalize the blood flow in an ischemic limb to the controlled non-ischemic limb. We make the limb ischemic by doing a femoral artery ligation, and you can appreciate there's some small level of spontaneous return of perfusion in all the controls, but if we deliver the VEGF appropriately and we build these blood vessels, we actually get more blood flow to that limb. So we can build structures that actually have some function. Now, this is relatively straightforward. It's very easy to recruit single-cell populations. Many times, we may not need to recruit just a single-cell population. We might need to recruit several cells. So, for example, staying in the same vein of angiogenesis, this is another schematic of the process where we have endothelial cells, smooth muscle cells. If we use molecules like VEGF, we recruit endothelial cells, but these blood vessels are typically immature and not fully functional. If we want them to become fully functional, we also need to recruit the smooth muscle cells. And there are other growth factors like platelet-derived growth factor that are very important in that process. And once those two cell types start to interact with each other, you get matrix deposition and formation of a mature vessel. So this led us to propose a couple of years ago that we needed to start thinking about delivering sequences of signals to recruit sequences of cells. And I should mention, we can't simply dump all these things together at the same time, because actually they have opposing effects, and if you do, they simply cancel each other out. So we had to modify our material systems so we could do things such as shown down here, where we'd have, for example, an early release of VEGF to recruit the endothelial cells, followed by a delayed release of a second morphogen that would recruit the second target cell type. And if you do this, what you find is you can indeed have a significant effect on the types of blood vessels that you form. So this is some histology in one of these studies. And if we don't do anything, we have some low-level number of blood vessels. They're actually stained brown here. So you can see there's a number of vessels. They tend to be small. This percentage refers to an indication of their maturity. Now, if we deliver VEGF by itself, we build lots of blood vessels, but actually they tend to be small and quite immature. If we recruit the smooth muscle cells only, we don't build many blood vessels, but actually the ones that are there tend to be relatively mature. If we deliver VEGF to recruit the endothelial cells, followed by PDGF to recruit the smooth muscle cells, we build lots of blood vessels, but now they mature and they become, as I'll show you down below, more functional. So this again is looking at this hind limb ischemia model, in this case in a model of atherosclerosis in mice, so an APE knockout mice. So we eliminate about 90% of the blood flow. There's some spontaneous return in the control. If we deliver VEGF alone, so we have a lot of blood vessels, but we don't tend to get a lot of function. But if we deliver VEGF followed by PDGF and drive maturation, we get much more blood flow out of that network. So we have the capability then of starting to recruit cell populations. And I should also mention that we, not surprising, in collaboration actually with Bob Goldberg, we've looked quite extensively at the larger vessel adaptations of these structures. And what we find is that with the appropriate delivery of growth factors, we get remodeling of the existing collaterals and their adaptation to larger vessels over time, and that's part of the story here as well, though I don't have time to go into detail on that. So we can recruit cells. We've actually demonstrated this actually several years ago. So what have we been doing more recently? Well, we've been asking a question of, can we control the size of the zone of the tissue volume in which we're doing this recruitment? So for example, can we recruit cells to a larger tissue volume? If we're going from a mouse to a human, we're going to need to scale up eventually. On the opposite, we may want to really control and have cell recruitment only to a very narrow tissue region, very small location. So how can we do this? How can we start to control the size and either get larger or get very much smaller in pattern with more precision? So we've been playing around with this for the last couple of years. One approach we've taken to try to enhance the volume of affected tissue is to exploit something called the EPR, or the Enhanced Permeability and Retention Effect. Now, as many of you know, blood vessels that are mature typically are not very leaky, so they contain most of their components, and things like nanoparticles or microparticles don't leave very readily. However, what was recognized a number of years ago is in tumors, where you have a lot of inflammation, a lot of ongoing blood vessel development, you have many leaky blood vessels where they actually have relatively large gaps in their walls and large nanoparticles can escape. And this is actually the premise for a lot of drug delivery in the cancer field today. Well, what two of the postdocs in the lab, Lan Chao and Zhigang Cao, had proposed a couple of years ago is they said, well, if this EPR affects in cancer, where you have rapidly growing tumors, maybe it's also happening in ischemic tissues, where you have a kind of a frustrated attempt to drive angiogenesis and build blood vessels that's always going on in the periphery, even though it tends not to be very successful in terms of returning overall perfusion. And the context here was that when you have hypoxic cells, I mentioned earlier, you get upregulation of a particular transcription factor and upregulation of angiogenic factors locally that will drive this angiogenesis process. So they said maybe we can very non-specifically target any random volume of ischemic tissue with nanoparticles. So they took silicon nanoparticles, they fluorescently labeled them, and then they introduced them using intravenous injection into mice where you would have one limb that would be ischemic and one that would not. Now, what you appreciate is this is before the ischemic surgery. When you have an ischemic limb, they demonstrated that you get a significant enhancement of homing and then retention of those nanoparticles throughout the entire volume of the ischemic limb. And actually, as we scale up this size, this ischemic region, or scale it down, we find that the localization of nanoparticles correspondingly goes up or goes down. They then proposed to couple therapeutic agents, like some of these proteins I've been describing, on gold nanoparticles, and they demonstrated when these are injected intravenously, they will indeed home to the ischemic tissue volume, will drive angiogenesis throughout that entire volume, and will actually get significant impact in terms of return of perfusion. So we can recruit cells in large volumes with this type of distribution approach. Now, on the other scale, what about getting tight spatial control? What if we want to really restrict the region where we recruit cells and build networks of blood vessels? Well, Yuen, a PhD student who just finished up in the lab, asked this question a couple of years ago. Now, clearly, biology does this all the time. There's all types of intricate patterning that happens in many organisms, as shown in this nice image I took from a science article a few years ago. So this is something biology does routinely. But typically, biology does not do this through the action of a single molecule like I've been describing to date. It doesn't do it by just having one promoter, because when you have a single molecule that promotes a process, typically diffuses away, and you get fairly shallow gradient, it's hard to pattern. So instead, how biology typically does this is by having spatially separated promoters and inhibitors that are coming from different locations, and as they diffuse towards each other, they will cancel out each other's effects, and you'll get very tight boundaries where a process will either be inhibited or it will be promoted. So what Will proposed was to do something very simple and to use VEGF as a promoter of angiogenesis and antibodies to VEGF as an inhibitors and put them in distinct spatial locations and see if he could create this kind of patterning and really get tight spatial resolution in cell recruitment. The approach he took, don't worry about the math here, but just very simple. Reaction, this is a reaction diffusion problem. He would have a source of each of these molecules. They could diffuse towards each other. They could bind. They could degrade. And his goal then was to create temporally stable and spatially restricted angiogenic zones. And I'll skip through the details, but by modeling this, he was able to predict where he needed to put depots of the promoters and the inhibitors in a tissue to get spatial control. And I'll just give you a piece of data here to document that this is indeed something that can happen. So we went back again to the ischemic hind limb model. And if we first of all look at this bar, this is, we'll look at, let's say, the capillary density in the muscle. So when we deliver VEGF, as I described before, from the polymers, we get a significant density of capillaries that gets built up as compared to doing nothing. However, what I didn't describe before is if we look adjacent to the region where we put this polymer, at some distance away, you get a really large spillover effect, which oftentimes may be useful. But in this case, we would not be desirable. So you get a lot of VEGF activity and a lot of angiogenesis, even in adjacent regions. However, if he would have the inhibitors and promoters, and he would look at tissue regions that were less than a millimeter apart, he can see in the region where we presumably were going to have the VEGF activity, we get equivalently the same activity as if we deliver VEGF alone, but less than a millimeter away, there's absolutely nothing. So we get no cell recruitment, no angiogenesis. So you can start to have pretty tight spatial patterning with this type of approach, and we've gone on to show this in a variety of other contexts. It's actually kind of interesting what he also showed, though I'm not going to go into the details of it, is functionally you tend to actually get improved outcome in terms of, let's say, the necrosis that happens in these limbs. if we provide tight spatial control over where we drive this angiogenic process. So kind of the net story here is one can recruit cells very readily by having some quantitative understanding of the activity of these molecules and then having the right technology to get the right concentrations and gradients within tissues. Well, what about if we have some cells that we recruited, and let's say we brought them into a material, can we get them to leave, and does that do anything useful? Does getting cells actually out of the material at the end of the day, which is very different than the classic tissue engineering context, actually have some utility? Well, we decided to look at this in the context of stem cell transplantation. So as actually probably everyone in this room is very aware, there's many, many trials of stem cell transplantation. For example, in the context of cardiovascular disease, many, many different cell populations, typically bone marrow derived, have been infused into ischemic heart tissue, into peripheral arterial tissue as well. And the general conclusion or the general observation that's been made in a wide variety of different clinical trials is there is a very limited return of function from this approach. And it may be at least in part due to the fact that very few of the cells that you transplant with this approach actually engraft and are there for any significant period of time. So what we proposed several years ago was a very simple idea, which is to take materials, to place cells within these materials, and to transplant them. So we, in essence, create a depot for the cells within the material. However, unlike the classic tissue engineering approach, the idea was not that the cells would stay within the material for a long period of time. Instead, the cells would be protected over the short term from the environment. They would be induced to proliferate within the device, and we would then induce them to leave the device in a controlled manner. So you can think of this as being very similar to a controlled cell delivery vehicle, analogous to the controlled drug delivery vehicles that I was describing earlier. And then the hope here is that we get distribution of these cells throughout a large tissue volume and a significant enhancement in their biological effects. So, quite simple idea. Now, we needed to think about how we were going to design these materials, and we've looked at a variety of different aspects of this. One of the very simple variables is, well, how are the cells going to move throughout this device? Now we need cells to be able to move around, come out of the device. My movies aren't always playing here today. Well, there they go. So from a structural perspective, Prakriti Talia in the lab did some very simple studies using 2-photon polymerized scaffolds and showed, for example, that if you have relatively large pores, what you can probably appreciate is cells are able to migrate very readily and actually respond to chemokines very readily. But as you start to get to smaller and smaller pores, the ability of the cells to migrate rapidly is significantly inhibited. So we had some design criteria for how we needed to design these materials from a structural perspective. And from a chemical perspective, if you go back to the work of many people in the past, Doug Laufenberg and many others, obviously the adhesive interactions of cells with materials also is really important in controlling their migratory ability. So if we think about these polysaccharide-based materials that I described earlier for the protein delivery, they're actually not going to be very useful in and of themselves for what I'm describing now because cells don't attach to them. So to get cells to attach and to be able to migrate, we couple small synthetic cell adhesion peptides, typically derived from fibronectin or other extracellular matrix molecules, as side chains. And I don't want to go through all the details of this today, but I do want to point out that similar to the growth factor delivery, this is actually a really rich variable space. So we oftentimes think about either having the peptides there or not having them there, but actually the context is equally important. So for example, if we think about this idea, we have a polymer chain. We can couple cell adhesion peptides, and just the degree of substitution, so how many there are, may be very important. Now these polymer chains fold in a certain way within the material, and so you're going to get some valency issues and some spacing issues at the nanoscale. When we make our gels, we may have some polymer chains that have peptides, others that do not, and so you're going to have spacing on another scale. And then obviously the mechanical properties of the gels presenting these peptides may also be important. And what we've shown over the last number of years is all of these variables can be exploited to get a really precise control over the behavior of cells within these materials. But for today, I'm just going to tell you we chose an appropriate presentation of these RGD peptides. And the first thing we did in terms of looking at cell release was to examine in vitro again, in a three-dimensional model, whether we could drive cells out of these materials. So we took porous materials that were built along these design criteria I mentioned. We present the right type of RGD sequence, density, nanoscale spacing, and then we placed endothelial progenitor cells within the device and looked to see would they leave. So if we actually don't have any mode for the cells to attach to the gels. And we place the gel within actually a collagen gel and just look for cells to leave, you basically see very few cells leaving. Now, if we have the RGD peptides presented, and we actually include VEGF, the molecule I was describing earlier, to recruit cells, and we put it inside the gel as well to make them more motile, we actually get some cells starting to leave. But actually a lot of cells stay trapped, which makes sense, because these cells like to go towards the VEGF, not away from it. However, we can take another isoform of that same molecule that doesn't bind the matrix, is freely diffusible, and doesn't set up gradients, so it activates the cells but doesn't really give them much directional information, and now the cells migrate randomly, and a lot of them end up coming out of the device. And this is just some quantification of that, and this was the work of Eduardo Silva, who's now on the faculty at UC Davis. What I'm not showing you here is if you continue this experiment, what you find is the cells proliferate within the device, and we actually get a multiplication. So we get many more cells coming out than we put in. Now, does this do anything? Well, Eduardo's looked at this a variety of different ways, and we're going to have some movies running down here, I guess. This shows you what happens if we don't do anything. We went to a very extreme model of high limb ischemia where the whole limb will actually necrose and auto-amputate, and so you can appreciate those animals are not terribly motile. Now, if we just inject the cells to mimic the current clinical protocols, and we include the same amount of VEGF as we do in the scaffold just to be consistent, or we take kind of a classic tissue engineering approach and we put the cells on a material that transplants them but keeps them, we again have some benefit here, right? So the limbs look a little bit better, but there still is a lot of necrosis, and you can appreciate that the mobility of this mouse is still not great. However, if we take the same number of cells and we put them on these materials that release them over time, you can appreciate here both limbs of this mouse look normal. It's going to go ahead and put weight on both. It's going to run around, and you'll see it actually in a moment. It'll actually stand up on its hind limbs, indicating a high level of functionality. So we've taken a scenario where you would get auto-amputation of the limb, complete loss, to one where we actually get a normal-looking limb and return normal mobility to this mouse. Now, we're able to accomplish this because we're building networks of blood vessels with these transplanted cells. This is just some histology showing that we actually get blood vessels if we do the bolus injection of the cells, or we do kind of the classic tissue engineering approach. But we get a density of blood vessels and a much more normal-looking structure of these vessels when we use this cell release approach instead of cell-keeping approach. And one way to try to quantify some of this is, again, to look at the amount of perfusion in these limbs. So again, if we do the ischemic surgery, the controls are not on here because the controls lose their limb, so we didn't bother to graph that here. But if we do the bolus infusion, in this case we're actually combining two different cell populations that arise out of these progenitors that are derived from the blood, either cord blood or peripheral blood. You get a little bit of salvage here and a little bit of blood flow. And if we use either cell type alone from the scaffold, we get a significant benefit. And if we deliver them together, we get 100% of blood flow. So it looks like using the cells and releasing the cells can give us, in certain situations anyways, tremendous effectiveness and enhancement in the ability of these cells to modulate regeneration. We've looked at this in a variety of other contexts over the last number of years, and I'm not going to go into all the details, but just one more example here. For example, in the context of myoblast transplantation to try to treat skeletal muscle injury. We show that if you do a bolus injection of cells, as is routinely done, this is actually the explanted tibialis anterior muscle, you get some regeneration, but if we take the same number of cells and use these materials as a bandage and have the cells slowly released, we actually get substantially more regeneration, as indicated here, simply by the significant enhancement in size of that muscle. If you actually look at the muscle fibers, we get active regeneration. When we get the release, we get lots of large muscle fibers that are forming, an active regenerative process, as versus the really stunted regeneration you see with the typical bolus delivery. And we get rapid resolution of the defect size when we do this material-based delivery. And though I'm not showing it here, these muscles actually show normal muscle function in terms of contractile function, while these have substantial deficits in functionality. So we can recruit cells, we can release cells. So now I'm going to talk about one more thing before we get into the bringing it all together, and that is in both these examples that I've been talking about, we're using what I would call conventional materials that do pre-programmable release. So we put things in the device, whether it's a cell or whether it's a protein or some other molecule, And it's designed so that that is going to come out at some rate that's predetermined before we put it in the body. Typically, it might be the migration rate of a cell, might be the diffusion coefficient of the molecule, it might be the degradation of the material. So, and that's what we see very commonly in a lot of materials in the field. What Johnny Zhao, who's now on the faculty at Duke, asked was, well, maybe there might be some situations where we'd actually like to have more or less real-time control. So could we have active porous materials for on-demand delivery? And how we proposed to do this was to incorporate small nanoparticles within the gels that would be responsive to magnetic fields. So in essence, ferrogels. So he took iron oxide nanoparticles, would place them inside the materials as we fabricated these gels to form the types of scaffolds that I've been showing you we've been using for the cell release and this drug release. Now, the key advance here was that these materials he was making to be highly porous. So you're probably familiar with classic ferrogels. This is kind of conventional ferrogels. You apply a magnetic field and you get some body force exerted on the gel and you get some deformation, but you tend to get very small deformations. By making these materials highly porous, though, what Johnny was able to demonstrate is now you can get extremely high deformations of these materials due to collapse of the pore structure. So much more, the response relative to the magnetic field is much greater. The reason for this is really simple from a material science perspective. We've now taken materials that were quite rigid and actually dramatically weakened them and made them much more deformable. However, they are also now able to undergo much greater compressive strains before fracture as compared to the nanoporous gels. So the hypothesis then was a very simple one, which is that this pore collapse and water convection can trigger cell and drug release on demand. We can place cells when we want them to come out. We simply apply a magnetic field. So Johnny actually demonstrated this in a couple of different contexts. I'll just show you one example here where he's taking cells, putting them on these materials that have these adhesion ligands to which the cells attach. He'd apply a magnetic field and he'd see, can he in essence pull the cells off the material and get them to leave. He first showed this in vitro, where we're looking at the percentage of the cells released as a function of time. In each one of these steps is application of a magnetic field. And the difference between these steps is how adhesive the gel is for the cells. If we make the gel more adhesive, then you release fewer cells than if the cells are less adhesive and basically can be removed more easily. And this just shows that the cells that are removed are indeed viable. And he's demonstrated that a variety of different ways. He further showed that we can do this in vivo. So we can take cells, we can put them on these devices, we can transplant them, and then when we apply the magnetic stimulation, we can in essence wash out all the cells on demand to get them out when we'd like. So we now have some ability to do on-demand release of these molecules in these cells and actually cycle some of these things or sequence some of these things. Okay, so now I'm going to switch gears and move on to the last part of the talk and describe bringing all these ideas I've been describing together. So can we recruit a cell, do something to it, get it to leave, and get something from it? And I'm going to talk about this not in the context of regeneration, but I'm going to talk about this in the context of immunology, and in particular in the context of new therapies for cancer. And I think all of us are familiar in this room with the current therapies for cancer, things like surgery, chemotherapy, and radiation. And clearly all these have significant limitations. And that has spurred interest in instead promoting a potent immune response or immunotherapies where immune cells in our bodies would attack and kill cancerous cells and destroy tumors. Now the basis or the theory underneath this idea generally is that cancerous cells are different enough from the normal cells in our body that our immune cells should be able to recognize them. So these cancer cells are antigenic, but oftentimes this does not happen because the tumor environment is immunosuppressive and prevents the immune cells from effectively interacting with those cancer cells. Now, if you could overcome this and have potent immune cell responses, this offers a variety of potential advantages as contrasted to current therapies. It includes the fact that immune cells traffic throughout our entire body, so you could have a systemic treatment. Immune cells have memory, so you could have the potential of preventing recurrence. And then finally, since we'd be targeting what is different about the cancerous cells as versus normal cells, you have the potential for minimal damage to normal tissue. So a number of potential advantages if this can work. Now, the key cell here that one needs to target if one wants to generate a potent immune response against the cancer is a cell called the dendritic cell. And this is just an SEM image of one of these cells in vitro. These cells you can think of as being the sentinels in the immune army. In an immature state, they traffic around throughout all the tissues and organs in our body, looking for what's there. And if they encounter foreign agents, for example, bacteria or viruses or other agents that don't belong there, these cells will become activated. They'll process little pieces of whatever they find. They'll present them from cell surface receptors, and then they will traffic to the lymph nodes. And once they're in the lymph nodes, they'll interact with naive T cells, which you can think of as being the soldiers in the immune army. And if these T cells get appropriately educated, they will then go out throughout the body and look for the molecular signature that they were trained to identify, and then attack and kill anything that has that molecular signature. So this understanding enabled the development of new vaccines, therapeutic vaccines, based on dendritic cells. And the approach to date, clinically, has been to overcome this immunosuppressive effect of the normal cancer or tumor environment to create a new environment by taking the cells out of the body. So the idea is you isolate dendritic cells from patients, you multiply them outside the body, you activate them, you give them what you want them to recognize, and then they're reinfused back into the patient. And the hope is that a sufficient number of these activated cells make it to the lymph nodes to generate a strong T cell response. Now this approach has been examined clinically. A company by the name of Dendrion received FDA approval recently for the first of these therapies. And what they showed was, on the one hand, quite impressive. They showed that this strategy led to about a 4-month extension of life. So that's obviously a significant benefit. But if you have cancer, you would like to get more than 4 additional months. So obviously the effectiveness here is something that you'd like to really improve. Not surprisingly with this approach, it's very high cost and very complex technology and regulation. So what we recently proposed was, going back to the slide I showed you at the beginning, to take our approach of in situ programming of cells and use it to address this particular issue, where we wanted to recruit dendritic cells, program those dendritic cells, and then send them to the lymph nodes and see if we could accomplish everything that's currently done ex vivo, but do it all in vivo with a little piece of plastic. So that was the goal. So these cells that I'm talking about, these dendritic cells, we're not trying to get them to do something they don't want to do anyways. These cells live to move around and to sample their environment. And this is just a movie showing these cells crawling around in a tissue culture plastic dish. And actually all the work I'll describe next was done by Omar Ali, a very talented PhD student and postdoc in the lab. Now, these cells want to move around, so the question just is, how do we get them to come where we would like them to go? Well, there's a variety of different drugs, including FDA-approved drugs such as GM-CSF, the trade name Leukine, that are known very potent attractors of these cells. So if you take these cells and you expose them to a gradient of this molecule, you can actually recruit them in a dose-dependent manner. So pretty straightforward, you can get these cells to come with exposure to this factor. Now, one of the things that Omar appreciated that had not been appreciated was, while this molecule very effectively recruits these cells, one of the other things it does is, when it's present at a high concentration, it actually inhibits their activation. And they actually don't express receptors that they need to be able to traffic back to the lymph nodes and actually talk to T cells. So the approach that he decided to pursue was to take this drug, place it in a polymer, and have it released for a short period of time to recruit the cells, and then the system would be exhausted, the concentration would fall, and the cells could become activated and could leave. So we took the most widely used biodegradable synthetic polymer, polylactides coglycolides, that many of you are very familiar with, and we used borrowed a technique we had developed a number of years ago to make highly porous materials. It was a technique we borrowed from the microcellular plastics field, and we wanted these materials to be highly porous so a lot of cells could crawl in and live inside this device for some period of time while we appropriately educated these cells. Now, if you incorporate GM-CSF into these devices and you place it in a rodent model, here we've placed one device on this side of a mouse that is releasing GM-CSF and a control on this side that did not. And this is a transgenic mouse where we can image the dendritic cells. You can appreciate this hot spot, meaning we recruited a lot of dendritic cells to the GM-CSF site. And we can quantify that, and you can see we have a dose-dependent recruitment of these cells to the site. Now, what Omar showed is we could recruit these cells, they could pick up antigen, and they could traffic that antigen back to the lymph nodes. So in this case, he used just a fluorescent tag that he put on the scaffold. And so we looked at the lymph node for cells that had fluorescent tag, meaning there are ones that are material and were also dendritic cells. And what he found was that at early time points, you actually had very few cells that came to our scaffold. It's this dotted line that came to our scaffold that made it to the lymph nodes, because they're actually all trapped early on because they can't get activated. However, once the GM-CSF concentration fell, which we measured, then the cells could become activated, and then the cells from our scaffold could then move to the lymph nodes and presumably talk to T cells. So this led us to then incorporate cancer antigens into the vaccine, to vaccinate mice, and then to introduce B16F10 melanoma cells, which is the standard preclinical model for melanoma. So a very highly aggressive, poorly immunogenic cell line into these mice. And what he found was, on the one hand, kind of promising. These mice, if you don't do anything, they all die in about 2 to 3 weeks. Well, actually, this is showing tumor-free. They would be dead about a week or two offset from formation of tumors that are palpable. Now, if he optimized the formulation, what he found is about 25% of the mice in this context would actually destroy all the cancerous cells and would go on to live a normal lifespan. So that sounds promising. We can take a situation where everyone dies to now 25% live. However, the approaches where you manipulate the cells outside the body, as I'll show you in a minute, have a success rate about 50 or 60%. So this looks promising, but no one would ever use it, right? You're not going to use an inferior therapy just because it's a little bit cleaner and cheaper. So this led him to go back and say, well, what am I missing? Why am I not getting a better effect? And what he proposed is that he wasn't doing a very good job of programming the cells, actively programming the cells inside the device. And he also wasn't doing a very good job of getting a lot of cells shuttling through the system. Now, what I didn't mention to you earlier is when these dendritic cells are trafficking around in the body and they pick up parts of foreign invading agents, they also get activated by having a general recognition mechanism to parts of things that, organisms, for example, that routinely invade our body. And these things are called danger signals. They're things like the membrane on bacteria, types of or chemical sequences in bacterial or viral DNA that is distinct from that found in mammals. And these dendritic cells have cell receptors that can bind these different fragments, regardless of, for example, what kind of bacteria it might come from, and they activate the cell and tell it that whatever you're seeing with this is something dangerous. So what Omar proposed, what we really needed to have at least 2 distinct signals in the system. We needed this GM-CSF that would create a gradient in the surrounding tissue, draw the cells in. But once they came in, we needed to have this danger signal to activate the cells efficiently so they could then move back to the lymph nodes. So the approach he took was to synthesize small fragments, small oligonucleotides that mimic bacterial DNA, basically CpG-containing oligonucleotides. We actually condensed these with the polyketone, polyethyleneamine, to make small nanoparticles. And the reason for this is that once you take this small oligonucleotides and make them into nanoparticles, shown here in this EM view, we found that cells take them up much more efficiently. So here we're looking at cellular uptake of either free CpG or condensed CpG. The condensed gets taken up much more readily by these cells. The PEI is mildly inflammatory in and of itself, which sometimes is considered a disadvantage. Here it's actually an advantage because you're actually trying to create an inflammatory environment. Now, really strikingly, if we looked at what happened in terms of activation of dendritic cells, first in vitro. So this is a receptor that cells need to traffic to lymph nodes. And as I mentioned earlier, if we expose cells to increasing concentration of GM-CSF, you actually get downregulation of this receptor, so they can't make it to the lymph nodes. However, once these cells are exposed to CpG, you no longer have any inhibition due to the GM-CSF. So they should be able to get activated and be able to traffic. And actually, if you look at the ability of these cells to migrate towards a chemokine that would be coming from the lymph nodes, if you have the unstimulated cells, meaning no CpG, as you expose them to more and more GM-CSF, their ability to migrate towards the signal is significantly diminished. And I apologize, this actually doesn't show up very well. But if we expose them to condensed CpG, their migratory ability is intact. So this led Omar to go back to that study I described before and say, well, what happens now? Do we get more cells trafficking through the system? One of the first things he noticed is if he looked at the lymph nodes of these mice when he explanted them, is with these infection-mimicking polymers, the lymph nodes are markedly enhanced in size. So when you get ill in the wintertime, you probably notice your lymph nodes get larger, and that's because you have more cell infiltration. So that's what we're seeing with these infection-mimicking polymers. And this shows the control, which is quite a bit smaller. If he actually quantified the number of cells that came to our material, picked something up, and then left, and were in the lymph nodes, you can appreciate by 2 days, he's already having substantial numbers of cells that have trafficked through the device, and by 7, he's starting to have hundreds of thousands of cells that have gone through the device. So one way from a chemical engineering perspective, for those of you who come from that field, what we've done is we've taken what was in the first iteration a batch process, where we brought in a group of cells, we manipulated them, and then released them, and we've transitioned that into a continuous process, where we've looked at these cells, we bring them in continuously, they live in the device for about 6 hours, and then they leave. And so we're shuttling a lot of cells through these devices. Now, we wanted to go back then and look at how effective this was then at generating an immunologic response against cancer. The approach we've taken is to incorporate antigens from a biopsy of the tumor, freeze-dry the powder, and incorporate it into the device to provide the antigens. So the first study that Omar then did was he went back to this preventive approach. We vaccinated the mice, he waited 2 weeks, and then he introduced the cancer cells. And he now got a very different picture than he did in the earlier study, where here we're looking at survival as a function of time. So if you don't do anything, all the mice die in a few weeks. If you have the cell-based therapy where you manipulate the cells outside the body, about 50% of those cells survive, or 50% of those animals survive, but now we get 90% of the mice with these infection-mimicking polymers that completely fight off these cells and destroy them all. And actually, I don't want to go into the details of it here, but it actually is important that the cells live inside the device. If we simply provide these biologic agents in various ways, without the cells living inside the material for a period of time, you don't get anywhere near the benefit. Now, I don't imagine that a lot of people here are heavily engaged in the immunology aspects of this, so I don't want to go through a lot of the details, but we've been investigating why this approach works. And there are a variety of different types of dendritic cells that are present in the body that have distinct functions. And one of the things that we've noticed with this approach to vaccination, and we think part of the reason this is so successful, is that we actually have very large numbers of CD8 dendritic cells that are very involved in cross-presentation, and then plasmacytoid that are very involved in generating a potent T-cell response. And we actually can find a very strong correlation between the presence or the numbers of these cells that we're able to recruit and the survival of the mice. We actually don't see any correlation between the numbers of conventional DCs, such are used in that dendrion product, in survival, which suggests that even if they transplant more cells, they may not get a benefit. Now, with this approach, I wanted to mention we are getting a specific T-cell response. We see antigen-specific T-cells in the spleen for extended periods of time with single vaccinations. So we're not just getting the DCs making it to the lymph nodes, we're actually generating significant numbers of T-cells. And equally important, though not an emphasis for today, there is another type of T-cell called the regulatory T-cell that downregulates immune responses. And what's been very frequently found in cancer vaccines is you can upregulate what are called effector T-cells, but then the tolerizing arm of the immune system shuts it down and you lose the effectiveness of your vaccine. What we have found with this approach is we actually do not upregulate the T regulatory response, we actually inhibit it. So we kind of turn on the gas and we knock out the brake and we generate a pretty potent immune response. So, does this then have a potent effect? Well, most times people are not being treated for cancer prophylactically. They're being treated therapeutically. You come and you already have a tumor. So the question is, can this actually have some effect? So to look at this, we gave mice cancer, and then we vaccinated them, and we looked to see whether or not we could prevent tumor growth. And if you don't do anything, you have the typical exponential growth of the tumor. If we introduce the cells that have been manipulated ex vivo, we have some benefit, and this was sufficient to move this into clinical trials, actually in phase 3 clinical trials today. So this is actually considered a very potent effect. But if we vaccinate once with our material, we actually drop the curve all the way to here, and if we vaccinate twice, we actually drop the curve all the way down to here. And what we're doing is not just slowing tumor growth. If you actually look at the survival of these animals, what you find is half of the mice that have been vaccinated twice have complete regression of established melanoma, which was at the time an unprecedented finding and still actually the only system that's been able to, vaccination system that can show regression of established tumors. So when we started going down this path, our hope was that we could do as well as the ex vivo cell manipulation by moving the biology into the body. Instead, what we're finding is by moving the biology into the body and orchestrating with materials, we can actually do much, much better, at least in these preclinical models. So that has been very promising. Of course, it raises a variety of questions, such as, is this a one-trick pony? Is this only good in melanoma? Could it be broadly useful in other types of cancer? We've been looking at this in the context of glioblastoma, and in therapeutic models, we actually see even greater efficacy in glioblastoma than we did in melanoma. We can begin to check the memory aspect of this, where we go back and re-challenge mice that we've cured once and reintroduce the cancerous cells, and we find that we have a very effective immune response when we re-challenge, and about 70% of the mice that are re-challenged completely destroy these re-inoculated cells, indicating we do have a potent memory response. And though I'm not going to go into details now, we also show systemic effects. For example, if we look at a model of breast cancer that normally metastasizes very significantly to the lungs, when we vaccinate, we can dramatically reduce metastases, as indicated here qualitatively and here quantitatively. So I'm going to stop here and just make a couple of quick comments. What I hope I've been able to give you a little demonstration is how we're trying to develop materials to what I would call programming cells in situ. And the data so far suggests that this general approach can really increase the efficacy of signals that we use to recruit cells in vivo. We can dramatically impact the activity of transplanted cells, and in certain situations, certainly not all, but in certain situations, we may be able to bypass all ex vivo cell manipulation. And with this, this may actually help in some way to make cell therapies more effective and practical approaches to treating disease. And so I'll conclude here, and if there's time, I'd be happy to take any questions.
Thanks, Dave. That was incredible. I'm sure there are a lot of questions out there. I think we will go ahead and take a little bit of time to take a few questions if we have some. There's one over here. Can we start there?
Hi. Thanks again for the talk. I had a question about the tumor model you're using. Have you tried this in a spontaneous tumor model? You're using isographs in most of these cases.
Yes.
And with the spontaneous model, do you expect any alternate effects in terms of diminished efficacy of the treatment or adverse effects because the cells are more recognized as?
Yes. Yes. So we've not looked in spontaneous tumor formation models. We actually have, I should say, we have studies underway right now in a couple of the breast cancer models, most breast cancer models. We've not completed any of those yet. So at this point in time, we do not know what the efficacy will be. Our expectation, though, is that that should work reasonably well. The providing protection prophylactically is typically more straightforward than therapeutically. In therapeutic models, in essence, we have to break tolerance. It's already been established. So our hope is that it will actually provide a protective benefit prophylactically as well, but we'll have to wait and see. Yes?
Dave, fantastic talk. Very powerful ideas there. I have two questions that are related. One is, tumors are heterogeneous, and there are many cell types, and there are many things.
Yes.
And so when we think about sensitizing and presenting the antigen, so how do you pick which one? Do you take a biopsy, and how do you do that in one part? And second, once you mount a response, how specific is that? And have you looked to see if the activated cells don't go about setting up autoimmune things and stuff?
Yes. Yeah, so for the first part, in terms of the specificity of the response, the physicians that we're working with at Dana-Farber actually are very cynical about the potential of defined antigens to be useful in melanoma. So the approach that we're taking right now is patient-specific. We take a biopsy of the tumor. We actually freeze-dry it. And so we then incorporate in the material, so we're presenting a mixture of antigens that are patient-specific. And that actually, the first clinical trial will be performed with that approach to antigen presentation. Over the long run, it would be very appealing to actually have a more uniform antigen or antigen mixture that you could use, and that's something that we're beginning to investigate in the laboratory. We know we do get potent responses to defined antigens, but whether or not those can be identified for specific types of cancer is still open-ended. Let's see, I think the other part of the question was about if we go too far, in essence. So are we actually breaking tolerance to things that we don't want to, and perhaps inducing autoimmunity? That certainly is a significant concern. If we do that, it actually suggests the system is extremely potent, which is in itself may be a positive sign. We do see some small indications of that. When you look at these models, we see, for example, in the context of melanoma, we do get some depigmentation at the site of the vaccine implantation. which is in the case that we're getting some response. Again, the clinicians we work with actually look at this as being a very positive feature, because they typically, when they see that, it's an indication that they're getting a potent enough response. If we go too far, we start generating a more broad autoimmunity, which we've actually completed all the preclinical toxicity and biocompatibility studies. Those are now complete. We do not see any issues at any organ site, so it does not appear that we're generating a broader, non-specific autoimmune response, but that's something that we'll have to see as we move forward.
Very impressive. Looking forward, from
Yes.
a more general standpoint, what are some of the challenges that you see in the designing of materials for cell therapies?
Okay. So, one of them is we need to understand the biology very, very well. So, for example, this last part of my talk, we didn't go into immunology because I suddenly had an inspiration that I wanted to become, cancer bioengineer. I wanted to actually do this with stem cells. And I didn't feel that I understood enough about stem cells, stem cell trafficking, to effectively provide proof of principle. In the immunology area, in contrast, there actually is a tremendous body of knowledge that was available. So that's why we went in that direction. And it's worked out quite well. We're now trying to do the same thing in other areas. For example, can we recruit specific stem cell populations, or progenitor populations is a better term, in adult organisms, and do the similar type of manipulation. Sometimes we know some of the signals, oftentimes we do not. So I think that's one of the big challenges. We simply don't have enough biological information to really design the systems appropriately. There are others as well, but I'd say that's probably the single biggest.
Any other questions?
You showed some very powerful data in treatment of cancer from vaccines using a sophisticated approach. Can you imagine that approach being translated into a prophylactic vaccine, or is there something more constraints on cost?
Yeah, so when we started going down this path, this particular vaccine system, I was anticipating would be proof of principle. And then we actually would go back and re-engineer the whole thing and deal with a lot of these types of issues. It's ended up being potent enough that there is actually a very large driving force to move it into the clinic in its current formulation. And for cancer, that seems to be highly appropriate. I don't think it'll be an issue. However, we would like to explore it more broadly. So, for example, we now have versions of this using different material systems that are injectable instead of implantable. This device will actually be, it'll be a small incision. It'll be placed in the subcutaneous pocket. But we'd like to be able to inject that, for example, with a catheter instead. And we now have those versions and actually have similar types of data showing effectiveness of that approach. But since those are different materials, we don't have the body of preclinical data. We're still sticking with the other system. But we're moving towards it, a broader application. The key thing will be what came up earlier is if we want to make something that's broadly useful, what antigens are we going to be able to use? Because if we have to personalize it, then obviously it's never going to be something that would use for millions of patients. We're moving towards that, and there's a variety of different ideas we have for choosing antigens, but that's going to be, I think, a significant impediment to go from treating, let's say, thousands of patients to millions.
Okay, I think everybody's hungry, so we're going to wrap this up. But you started your talk by saying there was a very simple approach, but obviously these are very big ideas and with very powerful implications. So we do have a small gratitude of thanks, or expression of thanks that we want to give you.
That was beautiful. Thank you. Thank you.